Hello, today I will tell you about the discovery of the human genome map. In 1990, a major step was taken to map humanity, and it was discovered in 2003. The 3 billion letters that make up the genome, the genetic codes in a human, and 20,000 genes have been solved. Genome mapping of human beings is the biggest discovery made in medicine in the last 25 years. This discovery will guide medicine. Genome mapping is used to identify and record the positions of genes found on a chromosome and the distances between them. Genome mapping has been a critical starting point for the Human Genome Project. A genome map shows the important places in the genome of the organism to which it belongs. A genome map helps scientists find their way through the genome. Important places on a genome map may include the genes themselves or the regulatory short DNA sequences that enable genes to be activated and passive. Provided the foundation for genome mapping, whole genome sequencing, and the Human Genome Project. The sequenced DNA fragments can be aligned to fit the genome map, helping to assemble and compile the genome. Over time, as scientists learn more about a genome, the map of the genome becomes more accurate and detailed. Gens enable the segregation of proteins, the chemical substance that determines a person's life, from hair color, height, food, number to the diseases they will be caught. The disease is tried to be prevented by repairing the changed gene that causes the disease. The healthy one is injected instead of the faulty gene. Thanks to the Human Genome Map, researchers have gained important genetic information on many diseases, such as Alzheimer's, inherited bowel and breast cancer. Diagnosis and treatment of diseases will be easier. Diseases that cause the death of millions of people every year, such as diabetes, heart, and cancer, can be detected and prevented long before, and domestic and national drug production for use in treatments. The average life expectancy can increase when the mentioned probability of getting sick can be limited thanks to the genome map, and when genetic diseases that can be inherited from their parents are prevented. Especially in diseases where carriage is in question, when the genetically carried defect is determined, it can be prevented from passing to the lower generations. It is possible to stop and eradicate the spreading diseases by learning the cause thanks to the gen map according to the inheritance type. However, it may not be possible to completely control the disease in some cases because the formations called mutation in the human genome that affects genetic diversity can occur spontaneously without inheritance, that is, without passing through the parents. It has been determined that we have approximately 30,000 genes, and these have not only individual but combined effects. In terms of diseases, it is reported that the genetic backgrounds of approximately 6,000 diseases has been determined. The change that a disease will be identified before it occurs depends on the mode of inheritance of the disease. If multiple gene defects are effective in the development of the diseases, it is very difficult to detect and prevent them. Also, since the probability of getting sick is a multifactional event, the completion of the gene map alone doesn't eliminate this possibility. Only by ensuring that some resistance and repair mechanisms work genetically without error, the likelihood of getting sick can be limited. The gene map will facilitate understanding of bioarchaeology, anthropology, the evolutioning process, and historical migrations. In this way, we will be able to learn when and where people migrated from and with whom we are related. The detection of detailed genomic diseases will help the discovery of genes and sequences that play a role in disease etiology will be found. Fast and more specific diagnostic tests will be developed. New generation agents and drugs that target genes and consequently include disease-associated protein will be developed. It will be possible to repair or even remove defective genes through gene therapy. To produce a genetic map, researchers take blood and tissue samples from members of a family, some of whom have a particular disease or characteristic. Then the data in the sample taken from each individual is separated and examined closely, and attempt is made to find a template that others don't have in the DNA of individuals with the disease. These are called markers, and they are extremely important in tracking the inheritance of traits across generations in a family. Different genome mapping types. There are two general types of genome mapping, genetic mapping and physical mapping. Both of these genome mapping variants guide researchers to the location of a gene found on a chromosome. However, they provide this based on very different information. Physical mapping looks at the physical distance between known DNA sequences. It does this by calculating the number of base pairs between them. Genetic mapping looks at how genetic information is shifted between chromosomes or in different parts of the same chromosome during meiosis. In other words, it deals with the part change event. Thank you for listening to my presentation.